program of the day 3 is the kickoff through the closing keynote by our great colleagues, the Doug Engel. So Doug Engel will give us a closing keynote, and his title is here the something different, but he's changed the title and Denouement. I've never seen this word, but anyway, he is going to explain us what this word means. And the subtitle is How did medical science evolve over the last 40 years to devise and integrated learning health systems? And of course, you all know the Doug Engel. Doug is the most simple description is I did my postdoc in Doug Engel's laboratory when I was 28 and he was 35. So naturally, the young scientists are, fighting a lot. And so he's a real good friend of me. But also, he's a molecular biologist trained by Tom Maniatis, and the molecular cloning age of the scientist. Since then, he's been working various aspects of the molecular biology, and now evolved to the learning health system scientist. So, Doug, please.
Well, thank you for that interesting introduction, Masi. So, Chuck correctly pointed out that the title of my talk means that I can talk about anything that I want to. It's a completely open area. Denouement was the word for used in Greek tragedies for either one of the authors or one of the players in a play to come in and explain how all of the bits of a drama wrapped together to finally come to a conclusion. And that was what I was supposed to do for this symposium. But as it turns out, this subject is something about which I knew nothing up until about 3 days ago, other than looking at Chuck's YouTube video. And I've learned a lot, and I'm going to explain what it is I took away from each of the talks up to today. And so the misinterpretations are entirely mine alone. So I hope you'll bear with me. We're required to show this now at the University of Michigan. I own a tiny, tiny interest in a company that's attempting to develop a therapeutic to treat sickle cell and beta thalassemia. So I'm going to give little insights that are probably incorrect, and you can all correct me in the question and answer period, that I found interesting in each of the talks. So the first day, Dr. Arai told us about TOMO and how it was established to provide information technology support for the Tohoku region residents as an online community medical platform, and to attract young medical professionals back to an area that was underserved to analyze population-based and 3-generation cohort studies. One of the interesting things I found out in Dr. Arai's discussion was that the Japanese MEXT budget was of 25 trillion yen, or translated to about 200 billion US dollars, is in a country whose gross domestic product is about a third of the United States, and yet it is 7 times the NIH budget, which is an interesting commentary on where the two countries feel resources should be put. After Dr. Arai spoke, Dr. Friedman introduced us into a learning health system. And you've all seen these circles for the last 2 days. And it was basically an idea that Chuck had to a group of investigators would come together in a decision to study a particular problem. They would assemble the experiential data and analyze it, interpret the results, and then the key, oh, thank you. The key was to provide tailored messages to decision makers so that those data could be acted upon. And hopefully, those decision makers, if they were suitably convinced, would take action and improve a particular situation. Now, as we all learned, the learning health system circles are fundamentally a platform for analyzing, for introducing and analyzing, and then developing ways of communicating with our overseers what we've learned. And so in the platform, we have mechanisms for communities of interest to use, policies that govern access to patient data, which is critically important, technologies for aggregating and analyzing data, and this is something that we obviously determined is a strength here at TOMO. Technology and policy for making knowledge both persistent and shareable among a wide variety of different platforms, and mechanisms for tailoring messages to decision makers so that positive outcomes from these health experiences, learning experiences, could be shared and health could be improved upon. The way that Chuck visualized the learning health system to work is an extensive group of shareholders who all both put information into the learning health system and benefit by taking information out. And this can be universities or research institutes, hopefully the government public policy institutes, and a variety of other partners. Dr. Nakaya discussed the Tohoku Biomedical Information Highway in personalized medicine and disease prevention, and stressed the point that separation of data and the semantics used to describe the data is absolutely critical. One would hope that, as Dr. Cush discussed, that one might produce a standardized package for regional, national, and even international consumption. Dr. Cush then reviewed the importance of establishing and maintaining standards, and why are we concerned with that? Well, the meaning, standards can preserve the meaning of information after it's exchanged from one culture, one platform to another, and this was referred to as semantic interoperability. The quality can be maintained so that the standards ensure the quality of data when they're collected and then exchanged. And the traceability is also a critical aspect of established standards, because the standards can carry metadata for audit trails to ensure that the data exchanged retain their integrity. Once one establishes standards, it also aids in communication, because different shareholders or different partners can understand exactly what any particular standard is established for. and finally, of course, and importantly, to medical system standards save time and money. So she told us about two important groups. One is the CDISC, which she heads, the Clinical Data Interchange Standards Consortium, which has developed is a standards development organization that was founded almost 20 years ago, and attempts to establish standards among a huge variety of participants, some 350 now. And this has been, I guess, most successfully applied to clinical data acquisition standards harmonization, or CDASH. And these concepts and standards are going to be required by the pharmaceutical industry and by the United States Health and Human Services and FDA very shortly. Dr. Delaney next discussed Transform, which deals with the interpretation of information content from a variety of sources to foster the integration of electronic health records into clinical trials. He gave 3 examples, type 2 diabetes, esophageal reflux, and diagnostic decision-making for how electronic records could be used. He also addressed the challenges in developing the learning health system. Absences from records are hardly ever noted by physicians. Doctors use codes differently. There is inherent system heterogeneity, and contextual meaning and its potentially erroneous translation between platforms can be extremely problematic, if not disastrous. The aims of Transform are to develop methods, models, services, validated architectures, and demonstrations to support epidemiological research, research workflow embedded in the electronic health records, and decision support for diagnosis. Dr. Frizzy next discussed the Nashville experience, where a group of apparently not very nice competitors who really didn't trust one another learned that through cooperation, which they referred to as the Health Information Exchange, a simple health record system, they could demonstrate a positive financial impact for all participants. They also discovered in studying the frail elderly that the most productive interactions were often not between the patient and the physician, but rather an extended group of individuals from both sides. Dr. Kimura addressed issues regarding data recovery from health information systems in a standardized way. He showed how these data could be used, for example, to determine how specific geographic boundaries impeded hospital visits, or how different oncology clinic availabilities determine patient preferences. Further, he showed a very impressive demonstration of a rapid database search for how many patients out of 50 million reported negative responses to a specific prescription drug using a storage retrieval tool, a specific storage retrieval tool. Dr. Menegishi then discussed the Tomo Biobank specimen collection, validation, and quality control, and how, using the laboratory management information system, they have been able to estimate errors in cohort assignment studies. Dr. Nagasaki then discussed the outcomes and future goals of the Tomo 1K Japan whole genome sequencing project, both hardware supporting facilities and computational capacity. Very rare single nucleotide variants were identified among the 1K-JP and cohort, which should inform multivariate inherited trait analysis. Dr. Ogashima next discussed the Integrative database for Genomic Prospective cohort study here at Tomo. They've developed an integrated database platform drawing on omics, specimens, baseline follow-up, and clinical data. Dr. Curie gave a really beautiful talk, I thought, presenting studies on genome medicine based on the Tomo Prospective Genome Cohort. The study tested the hypothesis that genetic factors plus environmental influences generate disease onset in multifactorial diseases: autism, glaucoma, bronchial asthma, and atherosclerosis. One interesting example was an estimation of the incidence of rare Mendelian disease using the 1K Japan database. Polyketonuria, which is caused by homozygous mutation of phenylalanine something that begins with H, is known to affect about 1 in 300,000 Japanese by direct blood screening. The heterozygous frequency, known from 1K Japan to be 1 in 105 Japanese, predicts a homozygous disease mutant allele frequency in the population to be about 1 in 45,000, closely matching the actual number. So this is a way that we should be able to use in the future these genomic data to predict possible disease frequency. Next, Dr. Kuriyama discussed disease appearance after the Great Eastern Japan earthquake disaster through the 3 Tomo community initiatives, the population-based adult cohort study, the birth and 3-generation cohort study, and the child health survey. He provided interesting insights into survey limitations and strengths. Dr. Stolk described some of the experiments from the advanced database use of lifelines in the Netherlands. While they don't do research themselves, they do provide data worldwide with more than 200 approved proposals for access to their database, and more than 100 publications are credited to the Lifelines data. Lifelines has established 167,000 participants with a 30-year follow-up in a 3-generation design. One interesting study was why do some people age well and others poorly? And the data enable research on environmental, phenotypic, biological interactions with genetics. Examples of how they are addressing precision medicine are studies on pesticide and lung function, or alcohol intake and cholesterol level. And he gave us one final interesting example of income and depression. Dr. Tsai described the Taiwan experience under a single national health insurance program and how their biobank and sequencing studies were used to investigate and react to the very high incidence of liver cancer. The data were used to argue that all Taiwanese newborns should be immunized against hepatitis B from birth, which has seen the incidence drop, I can't remember if he said 3-fold or 10-fold, but significantly, a very successful analysis and public health outcome. And finally, I wanted to share with you some of the my personal observations about what came up in the presentations and what didn't. What came up in the presentations multiple times and was stressed by multiple speakers was that Tomo is clearly strongest on the blue side of the learning health system circle. That is, if it were to become a true learning health system, there needs to be better communication with overseers and more use or advocacy to the government people who determine what to do. A sure prognostic indicator for failure appears to be to try to accomplish too much all at one time. So a number of investigators told us that one of the ways that their smaller focused learning healthcare systems, or their systems, were successful was because they didn't try and bite off too much. They didn't try and develop the perfect system from the very beginning. And then finally, and very clearly, one needs to make sure that you thoroughly research and adopt the best and appropriate standards, or be sure to participate in establishing those standards. this appears to be critical if you really do wish to grow a learning health system and interact with other learning health systems. What wasn't really discussed from my perspective was that there were no milestones of what should be accomplished by TOMO in the next year, 3 years, or 5 years, which I think is important to establish measurable goals. And finally, from the Frizzy doctrine, what does TOMO really, really want to do? So the Frizzy doctrine, I lost it. So sorry. States that what is it you are able to do, what is it that you want to do, and what are you really, really good at that you love to do. So I have accepted this, and you know, I don't think Mark made this up originally. I've heard it in other ways from other sources. Did you?
It's a variant of Jim Collins, a goodie break.
Okay. I followed this doctrine unknowingly for my entire career. I love making discoveries and finding a new fact. I also love not having a boss, which I've never felt like I've had a boss the entire time I've been a scientist. Following the Frizzy doctrine, it appears to me that what TOMO has established itself and being really good at is the technology of generation of novel information. And perhaps what they have not fully elaborated yet is integrating that information with electronic health records. And that's a choice that TOMO is going to have to make. They've made outreach. They have established connections with physicians both locally and elsewhere in Miyagi Prefecture, and that may evolve, but it doesn't appear to have done so yet. So in summary, I'd like to share this vignette with you. 45 years ago, one of the great biological philosophers of the early days of molecular cloning, called Gunther Stent, a professor of molecular biology at Berkeley, wrote a treatise entitled The Coming of the Golden Age, a View of the End of Progress. His basic tenet was that due to the amazing acceleration of scientific discoveries in the 20th century, that we would very shortly reach a time at which no genuinely new, important new knowledge would be forthcoming. And mankind would basically exist in a kind of utopian Polynesia. He thought there was, with the exception of only a few small issues, nothing important left to discover after 1969. Since 1969, there have been a few small discoveries. Restriction endonucleases, DNA sequencing, eukaryotic genes are packaged into chromatin, oncogenes are mutant cellular genes, introns exist in eukaryotic genes, individual genes can be isolated from libraries, and this was really the basis of the genome projects. There's self-splicing and regulatory RNA. Enhancers regulate eukaryotic genes. PCR was first introduced, which we originally referred to as purloining competitors' reagents. Automated DNA sequencing was introduced at Applied Biosystems, and as we've heard, the price has dropped by many logs since this original discovery. And finally, the Human Genome Project was initiated in the United States, at least, originally by Jim Watson, and then taken over by Francis Collins and Craig Ventnor. And if any of you know these characters, it's very clear why there hasn't been a Nobel Prize awarded for the Human Genome Project yet. Epigenetics was really not founded, but discovered rediscovered by Shirley Tillman and David Alice. And somewhat later, Professor Stent, who made the remarks in 1969 that nothing new was to be discovered, said, oops. So the take-home lesson from that little story is that hubris is a terrible thing. We've discussed over the past 2 days is to a large extent an ideal or goal. Working toward that goal, TOMO will likely encounter delays, cultural and legal obstacles, obstructions that are both valid and invalid, bureaucratic limitations, and many other impediments toward achieving an integrated learning health system. All of these barriers could be recognized as unavoidable viewed in the context of history and change. The goal of achieving a transparent and regionally, nationally, or even globally accessible records database and learning healthcare federation will have as its greatest achievement better disease diagnosis and prognosis, better treatment, and more precise knowledge, empowering physicians everywhere to ensure that my newest granddaughter will have the best possible health care. So with that, I know. Let me say that I'm delighted to be here. The reasons being, when I left on Friday, as Chuck said, I had to pull over to the side of the road to take a photograph of the thermometer in my car, which registered at minus 23 degrees Fahrenheit. So this 70-degree temperature differential has been very pleasant. Thank you all.
Thank you very much, Doug. Great remarks, closing the great keynotes, and I'm very much enjoyed. Thank you very much. And this presentation is now open for discussion, and you can comment or claim or throw bottles or whatsoever. Not shoes, okay? Not to throw shoes. Okay. Any comments, please? Yes. Do you have any comments? Well, Doug, your comment on the TOMO and what TOMO want to accomplish 1 year after, or 3 years after, 5 years after, that's a very good point, and it's a crucial for us to think about. Of course, we have a blueprint and plan for those 3 years, 5 years, but probably with the knowledge and accomplishment of this symposium, we're going to brush up that one. Well, any comments? Chuck, do you have any comment?
Well, first of all, Doug, thank you for that superb synthesis of the meeting. It's always very important to have somebody, especially with my memory, remind me of everything that happened, because it enables you to put things together in a different and better way. And I think Doug really made that possible, not just for me, but I would guess for everyone. I will just comment that at the 10:30 session, I plan to rise a bit to the Frissy challenge, and with full humility, and hopefully I won't do what Professor Stent did. I will make some specific suggestions for what might happen in the next year, or 2 years, or 3 years. That will be exactly that, suggestions, and I'm sure my colleagues, Drs. Delaney, Frissy, and Cush, will respond to that, and then I expect everyone else in the room will respond to that, and maybe we can begin helping you on that journey, Masi, which you have just said you plan to take. So we'll see what I can come up with at 10:30, but Doug's talk was a wonderful start on that journey.
Great. Any other comments or questions, or? If not, Doug, thank you very much.